Hello, everyone. Today, I would like to welcome you to our webinar, QIAGEN webinar, on optimization of wastewater SARS-CoV-2 samples to enable NGS-based detection of emerging variants, which will be presented by Dominic, Director of Microbiome Product Development at QIAGEN, and Jonathan Shaffer, Director of R&D for NGS assay technologies at QIAGEN. My name is Kasia, and I'm a Global Campaign Manager at QIAGEN, and I will be your host and moderator today. So Dominic will begin the webinar by first discussing the challenges of working with wastewater and the preferred methods for viral purification. And he will be followed by Jonathan, who will discuss the QIAseq Direct SARS-CoV-2 enhanced workflow, which offers several advantages over RT-based SARS-CoV-2 NGS methods. If you have any questions during the presentation, please submit them into Q&A box, and we will do our best to answer them at the end of our presentation. Welcome to you both, Dominic and John. It's a pleasure to have you here to discuss this very timely subject. So we're looking forward to your talk.
Great, thank you, Kasia. So I'm pleased to be here to be providing this webinar. Before we start, a quick disclaimer that everything that we talk about in these webinars are intended for molecular biology applications and not intended for diagnosis, prevention, or treatment of disease. And as Kasia mentioned, this is our agenda. We'll be talking about the extraction technologies needed for wastewater analysis, and then the NGS workflow afterwards. So wastewater surveillance has become very important over the past couple of years. It's estimated that you could save up to $1 billion on monitoring programs by monitoring wastewater samples, so looking at entire sewer sheds rather than trying to do individual monitoring, as it gives you a very good sensitivity of detecting infection within a large population. And of course, it provides the opportunity to see infections prior to infected people coming in for testing, and give monitoring for places that would not otherwise have that monitoring, and be able to provide very large overview of what the actual epidemiological situation is. And in the context of this webinar, also provide the opportunity to find new and emerging variants by doing sequencing of these wastewater samples. So of course, how do we find viruses in wastewater? Viruses are generally shed from individuals through their feces into the sewage, which then gets collected throughout the entire sewer shed into the wastewater treatment plant. And at the wastewater treatment plant, of course, the sewage comes in and then goes through various treatment methods in order to purify it before it gets released again as cleaned water. Since we are attempting to find a potential pathogen, we of course would like to look at the influent prior to any treatment of the sample. However, this makes it has its own difficulties in that the influent is a heterogeneous and generally dilute sample, which requires us to do some amount of sample processing, specifically concentration. And there's different ways that you can do it. There's virus absorption, there's ultrafiltration, you can centrifuge the samples, you can do a precipitation with various methods. All of these are ways of concentrating the viral particles out of your raw sewage sample into a form that is amenable to further processing. And then when you process it, you need to do an extraction of all of the nucleic acids that are present in order to get your viral nucleic acids, which can be then used or can be then analyzed with either PCR-based detection methods or by sequencing, or some combination thereof. And so QIAGEN has a total workflow for this process, going from the collected sample through NGS and data analysis. Here is an example of some of the applications that are done for this, and the workflow that we are discussing specifically today is going from your collected sample, doing sample disruption and sample extraction with various kits that I will be talking about shortly, and then going into the NGS and data analysis. And I will talk in detail about this first part of this workflow. So there's a variety of kits that we recommend for the extraction from wastewater. I mentioned already some of the difficulties of wastewater is that it's a dilute and heterogeneous sample, but it's also a very inhibitory sample. It's a very complex mixture of substances. Stool is known to be an incredibly inhibitory sample and very difficult to work with without having negative effects on your downstream analysis. And so we have specially developed kits. All of these kits here with power in their name include a technology that we call IRT, inhibitor removal technology, which removes those inhibitors out of the wastewater samples, which makes it easier to process while giving the highest possible yields due to an efficient lysis and a classic bind wash elute. Some examples of the kits that we have are the AllPrep Power Fecal Pro and AllPrep Power Viral, which will give you RNA and DNA from wastewater and stool, biosolids, and other materials. The RNeasy Power Fecal Pro and RNeasy Power Microbiome kits, which are designed for RNA from stool and raw wastewater. And then we have two additional kits which are used sometimes for wastewater samples, the RNeasy Power Water Kit, which is used to enable detection in very large volumes of water, as is designed for filtered samples, and the RNeasy Power Soil Total RNA Kit, which is well suited for sludge samples, as it can sample a relatively large amount of solid material, up to 3 grams. And then finally, we have the QIAamp Viral RNA Mini Kit, which is not a power kit, and so it is suitable for mostly clarified samples, so swabs, UTM, VTM, or clarified wastewater, where you do not expect to have a large number of inhibitors. So I've been talking a lot about the importance of inhibitor removal. Here's an example of a paper released recently supporting the need for PCR inhibitor removal during the extraction kit step when handling wastewater samples. And just to give an example of why this matters so much, this is how the IRT that we have works. When you break open the cells, or when you homogenize your sample, you're also releasing lots of other substances into solution. And there are many substances, such as humic and fulvic acids in soil, which can get washed into the wastewater through groundwater, polysaccharides, polyphenolics from waste material from plants, bile, bilirubin, heme from stool. And all of these either bind closely to DNA, and so they will co-elute with them, or they behave very much like DNA, so they will also be purified through the process. And then in the downstream, they will have serious negative effects on your analysis method. Importantly, significantly decreasing the efficiency of any PCR method. And so as an example, on the right, we have a PCR trace of what would happen if you take the same sample and run it with the inhibitor removal technology and once without. So when it says minus inhibitors, the inhibitor removal technology has been applied to remove the inhibitors, and the plus inhibitors in green are the ones where the inhibitor removal technology was not used and the inhibitors are still present. And you can see the really dramatic effect that this sample has. When we use the IRT, we have a very clean sample. The amplification curve proceeds normally, comes up early, and goes through an exponential phase. And when we have that same sample also purified, but without IRT, the amplification curves come up much later, the efficiency is delayed, and we have a delta CT of 10 or more, indicating more than 1,000-fold decrease in detection efficiency. So here it's very important, and as an example of what it looks like in wastewater, I can also show this here. This is an evidence that our wastewater samples are inhibitory. Here is an example of taking wastewater from local wastewater treatment plants and extracting them, or sorry, concentrating them either by centrifugation to generate a pellet, by ultrafiltration to generate a retentate, or by virus absorption on a mixed cellulose ester membrane in order to create a absorbed virus concentrated sample. And those 3 concentrated samples were processed with the QIAamp Viral RNA Mini Kit, which does not include IRT, as I mentioned earlier. And you can see that the CTs are all very high, up to 45. With the Omicron mutate, there's some variability, but it's also significantly delayed. And all three of those concentrated samples give actual signal for when used with the all-prep power viral kit, or the power water kit in the case of the MCE membrane. And so that's, you can see here as well, a delta CT between the non-IRT kit and the IRT kit of 7 or more, indicating really robust removal of the inhibitors, which of course gives you then detection of the SARS-CoV-2 that you are interested in. And so I'm going to talk really briefly about the virus concentration methods, because we spent a fair amount of time looking at which the concentration methods were the most efficient. And essentially, we found that different sample types or different sewage plants can give different efficiencies depending on the concentration method. So here, as an example, we have 2 different wastewater treatment plants. One of them is a more rural plant with more farmland. And so there's more environmental input into the wastewater. And the first plant is a municipal plant. So it's urban, and it's a much higher amount of human sewage present, and also a higher solid content. And so you can see in the municipal plant, the number of SARS-CoV-2 copies found were much more dramatically present in the pellet, while in the general plant, it was distributed across the 3 different samples. So what we have generally found is that the pellet is going to be the most efficient method of finding SARS-CoV-2 in the widest range of samples, but every wastewater treatment plant is a little bit different, and different concentration methods need to be optimized for the specific wastewater. We've also seen that both the normal filtration through something like the MCE membrane, as well as ultrafiltration, captures SARS-CoV-2 signal. And here again, you can see that the pellet tends to be slightly better than all other samples. And also, we have recently released the RNA Power Fecal Pro kits. This is a 2nd generation kit as a follower to the RNA Power Microbiome kits, which are widely used for wastewater surveillance. And these new kits extract higher levels of nucleic acid from these samples and are now the primary choice for wastewater surveillance of SARS-CoV-2. And as an example here, I have the quantification of SARS-CoV-2 in a local wastewater treatment facility, looking at the QIAGEN and GT molecular workflow using digital PCR, compared to Zymo quick RNA viral extraction and Bio-Rad droplet digital PCR. And you can see that the RNA Power Fecal Pro and QIAcuity extraction finds significantly more copies of SARS-CoV-2 per mil of wastewater that's been processed. And so our final recommendations for viral RNA extraction from wastewater samples are to concentrate the sample, either through centrifugation to create a pellet, through ultrafiltration, or through mixed cellulose ester filtration, and then process them with, for example, the RNA Power Fecal Pro when handling a pellet. And then your sample is well prepared for downstream analysis. As a final note, I'd also like to say that all of these kits that I've been talking about, the sample extraction that has been described here, are automatable on the QIAcube Connect, which will process the up to 12 samples automated from the lysis step on, and it will standardize and give you reproducible results. All right, so in summary, we believe wastewater samples must be concentrated in order to get robust detection of your SARS-CoV-2. Our kits provide rapid and efficient lysis to release the nucleic acids from these samples. We have very high recovery of viral nucleic acids, particularly with the RNA and All-Prep Power Fecal Pro kits. These kits include inhibitor removal technology, which enables your downstream analysis by removing problematic inhibitors, and the kits are automatable on the QIAcube Connect for standardization. And with that, I am finished and we'll hand over to John.
Thanks, Dominic. So while Dominic handled the sample collection, disruption, and extraction side, what I'll discuss today is the NGS portion of the SARS-CoV-2 wastewater surveillance. So there are a variety of key NGS library prep features for SARS-CoV-2 wastewater surveillance. First off, the library kits need to employ robust chemistry that has been specially designed for low SARS-CoV-2 titer samples. Tied in with this, the kit needs to be able to tolerate varying quality RNA samples. It needs to facilitate robust cDNA synthesis from low copy number targets, and it also needs to maximize sample usage. Within the library kit for the PCR enrichment, you also must be able to find the needle in the haystack. Specifically, primers need to be designed to give the utmost specificity for SARS-CoV-2, and the primers need to be designed to accommodate varying SARS-CoV-2 lineages. And the last point with this is that the amplicon size needs to be tuned for low quality samples. So ideally, overall, the amplicon should be smaller that allow for compromised samples or samples that are not of the utmost quality. The last key feature is that the library kit should be high throughput and employ rapid workflows. Paramount is that the kits include unique dual indexes, and this removes the problematic index hopping while sequencing. The kit should also be automation friendly because you're expected to produce libraries from hundreds of samples. And speed-wise, you should be able to go from sample to sequencer in the same day. Looking at the enhanced QIAseq Direct, this is an optimized library kit workflow for SARS-CoV-2 wastewater surveillance. Here we'll go over in the bubbles an overview of the workflow, and then we'll go stepwise through the workflow in the subsequent slide. Starting with RNA isolated from wastewater, you then perform cDNA synthesis. One of the key features is there's no dilution of RNA, and we've also enhanced the RT chemistry to detect low abundance targets. For this direct SARS-CoV-2 enrichment, this has been developed to accept increased cDNA input. Classically, the carryover of cDNA into PCR is capped at about 10% of your reaction volume. And due to our chemistry enhancements, you're able to carry over more material into the PCR enrichment reaction, allowing you maximal use of your sample. Additionally, we use overlapping amplicon primer pools that have been designed with primal scheme, which is used for Arctic primer designs. The main difference is we've tailored this for degraded samples, which is very optimal when using with wastewater samples. Last point is, regardless of copy number, we employ a fixed number of amplification cycles. This means that you can produce libraries from all of your samples at one time. You don't need to bin your samples based off of your real-time PCR CT value. After enrichment, you perform a single bead cleanup. The protocols are optimized to work with plates, making this very automation friendly. Next up, you perform library amplification and indexing. We use unique dual indexes, and this is all in a high throughput plate-based format. And we have up to 768 unique dual indexes. So you can multiplex hundreds of samples together at one time. After a final bead cleanup, and you're ready for sequencing. Looking at what actually happens in the reaction, you start with your RNA. First step is then you perform random primed cDNA synthesis. The cDNA synthesis is split between two reactions, one per primer pool. The two pools are overlapping 250 base pair amplicons, and the amplification handles designated with blue and green are included already in the primers. What this allows is then subsequently direct indexing during the library amplification. And again, these are unique dual indexes. One final bead cleanup, and then sequencing is performed using a 2x150 paired end read structure. A little more information on the enrichment primers is this is a critical point for the strength of the direct SARS-CoV-2 library kit for wastewater samples. First off, the enrichment primers have TrueSeq amplification handles. So this eliminates many steps that are found in other workflows and enables direct indexing via PCR. I mentioned this already, that we've used primal scheme for the primer design. So this is the same as the Arctic primers, and the guidelines for design are listed below. One of the key features, though, that we've done with the direct SARS-CoV-2 workflow is that after testing, low coverage regions have been boosted. And when you look at the final stats of the enrichment pools, the amplicon sizes range from 175 to 275 base pairs. What we found in wastewater samples is that this smaller, on average, amplicon size, smaller than, say, 400 or longer, is very well suited for wastewater samples. Also, the amplicon overlap is approximately 50 base pairs. So between the two amplicon pools, you're able to capture all variants of concern by the QIAseq Direct SARS-CoV-2. All right, so how does the kit perform? First off, I'll show you data with ATCC SARS-CoV-2 control, and then I'll show you wastewater detection. So here we have varying copy numbers of SARS-CoV-2, from 1,000 copies to 100 copies to 20 copies. And the CT value associated with 20 copies is about 35, and the CT value is designated here in each chart by the yellow line. Everything is done in the background of human universal reference RNA, and we allocate only 125,000 sequencing clusters per sample. So you don't need many reads in this context. On the left, we have first the mapped reads to SARS-CoV-2. So regardless of whether you have 1,000 copies or only 20 copies of SARS-CoV-2, you have nearly 100% of your reads being mapped to SARS-CoV-2, demonstrating the ultimate high specificity of the direct enhanced kit. Critical for clade lineage assignment is having a certain percentage of bases with greater than 20x coverage. And often, many kits suffer on the very low end, so the 20 copy range or lower. And this is not what you see with the direct enhanced kit. So whether you have 1,000 copies or only 20 copies, you have nearly 100% coverage across the SARS-CoV-2 genome, enabling efficient clade and lineage assignment. Tied in with this of how we achieve this very robust coverage is the uniformity of the primers. So uniformity metric is looking at the percentage of primers that are have greater than 0.2x of the mean coverage. And again, whether you have 1,000 copies or only 20 copies, the uniformity is very, very high. So greater than 98% with 1,000 copies or 100 copies, and even at 20 copies, you still have uniformity of 85%. And what this suggests is that across the SARS-CoV-2 genome, you have very efficient enrichment of SARS-CoV-2. Looking in the context of wastewater, this is NCBI genomic sequencing of SARS-CoV-2 from wastewater. And specifically, this is a bio project. It is the Genome Tracker Wastewater Project. And a little bit of background here is that the samples were collected as part of the US FDA's Pandemic Response Project for monitoring SARS-CoV-2 variants in wastewater. And this is third-party data that can be downloaded from these following links. The biosample is the same between the two kits. So library kit N and library kit QIAseq Direct. And highlighting some of the points when you look at the two workflows, library kit N has unidentified reads of greater than 78%. This means these are not reads that are going to SARS-CoV-2. Of the identified reads, which was approximately 22%, only 20% were determined to be associated with SARS-CoV-2. So what you can see is in library kit N, most of your reads are going to off-target. On the other hand, with the QIAseq Direct, the unidentified reads are less than 1%. This again attests to the high specificity of the primer design, and also that the Chi-seq direct primers are smaller, the amplicons are smaller, allowing for efficient enrichment of SARS-CoV-2 regardless of your sample type. Of the identified reads, so you have 99.28% are identified, and all of them go to SARS-CoV-2. So again, when you're thinking of an optimal kit for wastewater detection, the Chi-seq enhanced kit is something that you should consider. And in summary here, going back to our key features. So key feature 1 for NGS library prep from wastewater is a robust chemistry specially designed for low SARS-CoV-2 titer samples. The second key point within this is that your PCR enrichment must be able to find the quote unquote needle in a haystack. And from that wastewater slide we just looked at, you can see that 99% of the reads are going to SARS-CoV-2. Point 3 is that you should have a high throughput and rapid workflow. So when you look at the SARS-CoV-2 direct workflow, the entire workflow takes only about 4 hours from start to finish. And in addition, this is automation friendly, and it can be multiplexed up to 768 samples at one time. So I'd like to hand this back over to Kasia to finish up the webinar.
Thank you so much, John and Dominic, for your presentation. So we actually have a few questions being asked. And if we do not manage to answer all of them, we will send you answers via email. So the first question is for you, Jonathan. Are there any special considerations that need to be followed when preparing NGS libraries from wastewater samples?
It's a good question. I think the key feature is, beyond what we discussed specifically for NGS library prep, is that your upfront sample collection and processing matters. That your quality of your NGS prep is directly associated with your sample prep side. And it's basically, if you have strong removal of your inhibitors, you have a strong chance for success of prepping very robust libraries from SARS-CoV-2 for wastewater surveillance.
Thank you, John. The next question is for Dominic. What is the most effective concentration method for wastewater samples?
Yeah, so I touched on that a little in the talk. We believe that the most efficient overall method is going to be centrifugation and processing of pellets, has been our experience. But in general, you're going to want to have a method that can deal with a reasonably large volume. We suggest starting with at least 40 mLs, and then concentrating that down to an approximate size of 200 microliters or less for extraction into the kits. So something that can provide a 200-fold or greater concentration efficiency is something that you need. As I said, we believe that the most efficient method specifically for SARS-CoV-2 detection is spin down of the solids and processing of the solid material that is suspended in the samples.
Great, thank you, Dominic. The next question is for Jonathan. What read allocation is optimal for NGS wastewater SARS-CoV-2 surveillance?
Yeah, that's also a great question. This is a question that's been asked a lot throughout the pandemic. And a very good starting point for wastewater is about 1 million reads or clusters per sample. With NP swabs, we can routinely go down to 125,000 clusters or less, with the idea that your CT values are often beyond 30, particularly with wastewater samples. You want to allocate a little more reads than, say, that you do for NP swabs. I would start with 1 million with the idea that you can always downsample bioinformatically when you're analyzing your data and see the sweet spot of read allocation. And then as you continue to go forward, you can adjust your read budget to match what's most appropriate to call all the mutations in your sample.
Thank you, Jonathan. The next question is for Dominic. How consistent is the SARS-CoV-2 level in wastewater?
I'd say that it's actually quite inconsistent. So if you have a standardized workflow, for example, the workflow that we showed today, and you do that every day, you should be able to detect trends very consistently, but the samples are going to vary depending on the total flow in the sewer shed. During times of heavy rainfall, for example, the samples might be more diluted compared to other times. And so one thing that many people do look at are fecal indicators. And so that may be something that you need to consider, whether or not using a fecal indicator such as PPMOV or crossphage or one of the others could be helpful in normalizing the data that you get off of the wastewater samples.
Thank you, Dominic. There is actually another question for you. Are there other applications for wastewater?
Besides SARS-CoV-2, yes. So wastewater surveillance had already been used for polio surveillance prior to SARS-CoV-2 becoming a major topic. And now that the infrastructure and the experience is in place, I think many people are looking for additional applications. And a fairly immediate one that has significant interest is antimicrobial resistance, for example. So I think other viruses, as well as sequencing for antimicrobial resistance, might be further applications in the future.
Thank you, Dominic. Our next question is for Jonathan. When choosing an NGS library prep kit for wastewater surveillance, what should I avoid?
My biggest take on what should be avoided from seeing results and talking with customers in the field is longer amplicon designs often don't have the best success. It doesn't mean that you don't have success, you just have a better chance for success with smaller amplicons. And we offer at QIAGEN both an Arctic-based product, the QIAseq SARS-CoV-2 primer panel, that has 400 base pair amplicons. We also then offer the direct kit. And when you compare the two kits with wastewater, the smaller amplicon, particularly with the enhanced RT and PCR chemistry, gives the best results.
Thank you, Jonathan. One more question for you. For the QIAseq Direct SARS-CoV-2 kit, what is the lowest sample CT value that you've successfully derived libraries from?
That's a great question. So the holy grail is being able to prepare libraries across the entire dynamic range of SARS-CoV-2 titers. We've been able to efficiently prep libraries with greater than 95% of the genome at 20x coverage down to CT values of 38, 39. So at that point, you have very low viral particles, meaning that you have just a few SARS-CoV-2 copies. So if you're looking for something that can truly find that needle in a haystack, the enhanced chemistry has been specially designed to grab those very low titer samples.
Thank you, John. I think we can tie things up here. And if you have any more questions, please drop them in the chat box or email us. The email is visible on the screen. Thank you again for attending our webinar today. I also want to thank Dominic and Jonathan for joining us today to present on this topic. At this time, I will close the session and wish you a great rest of your day. Thank you so much.
Thanks, everyone.
Thank you.